He has held teaching appointments at universities in Sheffield, UK, Perth, Australia, and Berkeley, USA. He has been a research manager in Australia's Commonwealth Scientific and Industrial Research Organization. He is currently the head of the Bioinformatics Division of the Walter and Eliza Hall Institute of Medical Research in Melbourne, Australia. Professor Speed was in Singapore in June this year. He was an invited speaker for the workshop on genomic profiling at IMS, my institute. But unfortunately, he did not get to deliver his lecture because he was quarantined after he was found to have sat close to an H1N1 infected person in the plane from Melbourne to Singapore. However, he has kindly agreed to write an article on his experience in quarantine at Aloha Loyang, which he described as more like in a prison than in a resort. This article will be published in the November issue of IMS newsletter, English. It is a very interesting article. I would recommend you to read it, but you have to find it first. We'll go to our website and you will find it. It is now my great pleasure to invite Professor Speed to deliver his public lecture entitled Keeping Afloat in the Deluge of DNA Data. Terry?
Thank you very much for those kind words, Liz, and of course for the invitation to speak. It's a great pleasure to be here again, outside the prison. Check out the rest of Singapore. So, there's my title, Keeping Afloat, and if you want to know who I identify with, it's these people here hanging on by their fingertips on the verge of being swept away. This picture was inspired, I think, by the mythical flood that preceded Noah and his ark, but it captures rather well the feeling. I didn't invent this word deluge. We'll see a bit later where it comes from. So, just switch this on and make it work. My talk is about DNA. How do you like my animation? So, I'm assuming that you're not all that familiar with DNA, and apologies for the people who are. There'll be a teeny little bit at the end that maybe you want to know. Okay, so trends in DNA. I'm taking a historical view. The devices that read DNA and the amount they produce, because that's where the deluge is coming from. I'm also talking about computer power. Everybody knows about Moore's Law, and Moore's Law has kept us going for a while. But believe me, it's going to fail us rather soon in this field, I think, and I'll give some evidence. But it's also about trends in the mathematical, statistical, and computational methods. Because as the data deluge gathers apace, we find increasingly the methods we used in the past are inadequate for the future. And so we're being driven along by the technology. I like this aspiration from the Olympics. I don't know if anybody can translate this Latin. But it means something, something, and something else. Any volunteers? Von B. Who have a volunteer in row one?
Yes, swifter, faster, and stronger.
Swifter, faster, and stronger. Well, in the world of DNA, faster, cheaper, and better. I don't know whether it would sound better in Latin. I did translate it, but I thought you wouldn't recognize it, so I left it in English. But anyway, that's what's happening with DNA. And, of course, faster and cheaper probably means more. So now we get a little serious for a moment. This is a form of DNA which I'm sure you're all familiar with, where there is a double helix, and they have the so-called backbone, which here is in these colours, and then you have these bases which are joining across, and that's kind of more or less molecular view. A slightly easier view is this cartoon version, which we can see that we have C paired with G and G paired with A. So, whoops, that's called complementarity. And one of the advantages of this is that if we know one of these strands, we don't have to write down the other one. It's predicted by the complementarity. If there was a C here, there will be a G there, C there, etc. And the reason we need to know this is because the unit of length along the DNA molecule is called base pairs. So these are paired bases. I'm not going to give you both of them. I'm only going to give you one each time, but we're going to measure length in base pairs. 1, 2, 3, 4, 5, 6, 7, 8. So there's 8 base pairs, which we would think of as CG, TT, TC, TA. So we wouldn't bother saying what the other one is. Forgive me if this is all too elementary. I'll try to lose you in a few minutes. Okay, so here is a whole lot of base pairs, except you only see one of them. So AAA, TA, TAT, and in fact, there is 1,000 there. If you do your arithmetic, there's 10, 20, 30, 40, 50, and if you count down there, you'll see 20. So that is 1 kilo base pair. In this talk, you have to remember all of these letters. Kilo for 1,000. And anybody know what comes for a million? I'm looking for some audience participation here. A million? Mega base pair. Thank you. You're going to get all the questions from now on. Okay, so we're moving on. Kilo base pair, that's extracted, of course, if you want to put the actual DNA sequence of any organism or any gene, you'll be listing these guys here. And although it's called base pair, you only see half of it. Okay, so I do need to remind a couple of people perhaps that chromosomes, which are inside nuclei of cells, if the cell has a nucleus, are in reality very long DNA molecules. So here's a cartoon where we see this arm of that chromosome unwound and unwound and unwound and unwound, and eventually you see a double helix. So chromosomes are typically very long DNA molecules. How long? Well, human tens to hundreds of millions. Okay, so the smallest human chromosome is about 50 million, and the largest is about 200 million. So the average is about 150. So that's called megabase pairs. And you need to remember these numbers: kilobase pair, kilobase pair, megabase pair, so that you'll appreciate the deluge when I show you some decent figures. These are peanuts. Okay, so 150 million for a typical human chromosome. Now, genomes, like our genome and the wallaby genome and the dog and cat genome, and maybe even a bacterial genome, but often not as much, are 1s to 10s. That is to say, the bacterial genome might be 1 chromosome, human genome is 23, and though they're paired. And how, well, we have 23 by 2, and when you do the arithmetic there, we get 3 gigabase pairs, 3 billion base pairs. Now we're starting to talk real numbers, but this is just the beginning. Okay, so 1 human genome, 3 gigabase pairs, and it's going to go on. Okay, base pair, kilobase, megabase, gigabase. You'll have an opportunity to remember the next 2 or 3 in this sequence shortly. But I want to start by saying, why are we doing all this? I've mentioned about the DNA sequence. Well, here's a sort of serious biological reason, that we sequence the genomes of organisms to reveal the common molecular basis of these. Because basic biology is the same for us, for bacteria, for yeast, for flies, and we learn about this a lot by sequencing their genomes and interpreting that sequence. H1N1 flu. Well, if they want to find out whether the version of flu that I was carrying, if I was carrying flu, which I wasn't, was the same as some other variant, then they would probably sequence the genome of the flu virus that is inside it. This is done very commonly with HIV to track transmission and so on. The thing that Lewis mentioned, which I've spent a lot of time trying to do, is find disease genes. And we often use DNA sequence to do this. Diagnostic purposes, it was mentioned today that I was involved in a certain criminal trial that involved blood, and I'm sure you all know very well. In fact, I actually gave a talk here at NUS a few years ago about DNA forensics. So forensic use, genetic use, diagnostic use, which really covers this flu here. That's testing to see if DNA variants are the same. We sequence it. To explore the bacterial world, I don't know if any of you are aware of this interesting application of sequencing, which is growing now, but you know, in our guts we have lots of bacteria. We have many more bacteria in our body than we do human cells. But what are they? Well, largely they're unknown. because to sequence them and identify them, we would have to be able to culture them, and it's believed that there are literally tens of millions of species of bacteria which are unculturable, which are out there in the world, in different environments, in our gut, in the soil, in seawater, in a cow's rumen. I heard a talk a couple of weeks ago about sequencing the genome of a cow, sequencing the bacteria in a cow's rumen, because cow's rumen is a very, very effective, shall we say, chewing machine, that you can give it almost anything from grass to concrete, and after a certain time it will be small pieces that are extruded out the other end, and the bacteria that do this job so effectively are largely unknown to science, but you can learn about them by sequencing what's inside the room. Okay, so that's, and there's much more, and I will mention a range of activities of sequencing when I get to a little closer to the substance. So how do we sequence DNA? The answer is, of course, we use machines. But initially we didn't use machines, we used PhD students. And, so I'm going to go back to the early days of DNA sequencing, where they were in about 1975, when they were run out on electrophoretic gels. And you had a different lane for each channel, and these are what are called audio radiographs. This radio labeled, and you read off the T column and the G column and the C column, and you can see here, if that's a T and that's a G and that's a C, this guy over here is an A. So, and you can see here we don't have very many base pairs down that gel. And if you want to sequence a human genome of 3 billion base pairs, you probably don't want to do it that way. That would be quite a lot of PhD students. And we'll try to put this on the right scale in a few seconds. So this was an early technique. It's still called, it was called Sanger sequencing. There were some more primitive methods before Frederick Sanger invented this method. But Sanger DNA sequencing for that 20-year period. Here is a picture of the first more or less automatic DNA sequencer, which had a gel, had a gel not unlike that inside it, but a little bit fancier, but it was automatic and things were a little different. And I've just got the sort of development through here. They all have these 3-7 numbers. This guy here is just a sort of jumped up version of that. This is a capillary sequencer, which are still being used by some people. And this all happened over roughly a 20-year period. And it's all pretty much the same technology, Sanger sequencing. And that era ended a small number of years ago. But what it produced were these pictures here. And I don't have time to tell you about it, but there's a lot of interesting mathematics and statistics went into reading off these bases from these, what you call electropherograms, which were basically reading the gels. It's just a slightly more sophisticated version, faster, more multiplexed, 96 typically capillaries rather than one slab gel. But now we're getting to the present. This is one of the first, or probably the first, widely used commercial next generation sequencer from a 454. This is Illumina. I was at the GIS this afternoon, and I think I was told they had 8 of these. Anyone from the GIS tell me, is that the right number? Do I have a GIS representative that can confirm? I think they have 8 of these, and this is the Applied Biosystems, the same company that made all the ones on the previous slide, called the Solid System, and I think they had 3 or 4 of those over the GIS. And this is Helicos, which, as far as I can see, Singapore doesn't have one yet, but there are not too many of them around. So these are the next generation guys that are here now. So the next generation is here, and I'll tell you in a few seconds how many gigabases these guys generate. And then on the horizon, we have a couple of other technologies, Pacific and Oxford, representing the next generation. And this is going to be roughly, let's say, 100 to 1,000 times more than we're getting out of this. So it is the deluge after the deluge we're in, and it may arrive as early as next year. Okay, so these are on the way. So-called single molecule sequencing. This is actually single molecule too, but it's not on the same scale as these guys. So, I'm putting this together with a little flowchart of sequencing technology growth. And along here I have decade 1970, 75, 80, 85, 90, up to 2010. And up here I have kilobase per machine per year, megabase per machine per year, gigabase per machine per year. You have to tell me what TB means. What's the answer? Terabytes, right? 10 to the 12th. 10 to the 9th. They're going up by 1,000 each time. P? Petabytes. Okay, so that's where we're going here, folks. Per machine per year. So you have this machine, you run it sequencing data for a year. Now down here, sequencing by PhD. 50 to 1,000 bases per PhD. Okay, so that's around about 75. Then the Sanger sequencing that I showed you, those 4 versions. 150 kilobase per machine per year, 10 megabases per machine per year. When the automation came in, about 85, and the capillary one lifted that up to about 400 megabases per machine per year. Well, a lot of these machines were what's used to get us the first human genome sequence, which was 3 gigabases. And the company that did this, the so-called private genome project, I believe had something of the order of 300 to 400 of these machines sitting in a kind of a factory environment, each spitting out their 400 megabases per year. And if you do the sums, you'll get to 3,000 megabases pretty quickly. Next-gen sequencing is where we are now. And I was talking sequencing by synthesis. It says there 11 gigabases per machine per year. That's probably an underestimate at this stage. And, well, so, yeah, here's today. 5 terabases per machine per year. So, with, yeah, I don't need to tell you, terabases, 10 to the 12. So, if you have one of these machines running, you're going to generate terabases of data. And that's, here, you can't even predict what it's going to be by 2010 or future. So this is what's happening to us. So how much data do we have now? I can see there's a lot going to come. Well, go back to the same sort of timeline. And here, kilobase, megabase, gigabase, terabase, and petabase. And now this is not quite the total in the world, but the total that are in the public databases. So anybody who is publishing a paper is required to submit their sequence. And GenBank, which was the first public database, was created in 1982, and that's sitting there. And you can see that we've had, I didn't point out before, but we've had roughly exponential growth. And that's the case here as well. And I've just put in a few timelines here to show, there's the human genome up there at 3 gigabases, officially finished in 2001. The bacteriophage 5X174 was the first quote-unquote organism that was fully sequenced, and that's about 5 kilobases, 5,000 base pairs long, and then you can see a human mitochondria is 16,569 base pairs long, and then bacteriophage lambda is about 50,000 base pairs long, and Epstein-Barr virus is about 120. So sort of over these years, we're getting up bit by bit. H-flu was the first so-called free-living organism. This is not the influenza virus, this is a bacterium called Haemophilus influenza, and that is 1.8 megabases. Wow, we're over here. Fantastic. So that was in 1995, made a big splash. And now we've got yeast and the worm and fruit fly, and a human up here at 3 gigabases. So that's where we were, and if you do the arithmetic, that's a 150,000-fold increase over 26 years, which is slightly slower than doubling each year. And if you believe Moore's law, that's sort of supposedly about doubling each year, then you can see that we're keeping up with Moore's law. So probably the amount of data we generate, we can analyse on our computer, and we can store in our hard drives, because they're growing at about the same rate. And that's what I'm going to suggest to you is about to stop. Okay, so that's where we are now. So, the public next generation sequencing, in 1 year, that went up 70-fold. And, well, as you can see, it's kind of off the line. And we probably have too much data already of this next generation sequencing to be seriously analysed. And that is without the next generation, which I'm telling you is maybe a year away. Okay, so we're well off the exponential increase. We're barely coping with that jump, and if this, let's say 2011, might be up here somewhere, and we'll have to start doing log scales, and we'll see in a second that we really do have issues of data storage and data processing. But I want to show you this slide first, which came out in a paper last month in Nature Biotech. So, comparison of the estimated cost and effort in published human genome sequences. And I already mentioned that the first human genome sequence was published in 2001, and it was done by Sanger on these ABI machines that I showed you. The estimated cost, this is rough, is about $300 million. The paper had 251 authors. And it's, well, about fourfold coverage, more or less. And then this is, one of these is the public, and this is the private one. Well, it was about $100 million. This is Craig Venture's company, also Sanger. And they, I didn't tell you how many machine runs. Nobody knows how many runs went into the public database, but it was undoubtedly many hundreds of thousands. Of course, if you're looking down the column very quickly, you'll see a few things have changed, but I want to take you there slowly. So, the Venture show was estimated to be about 100,000 runs on one of those Sanger machines. Paper has 274 authors, fivefold coverage. A couple of years ago, this was revisited with another Sanger sequence, only cost 10 million, only 100,000 machine runs. So we're, pretty much the same there, but a much shorter paper and better coverage. And now we're into the next gen sequencing. 2008, a $2 million effort, which is an estimate of how much it costs to sequence James Watson on in a Roche 454 machine. The 234 runs, 27 author paper, 7-fold coverage. Let's cut to last month. The Holocaust machine, $48,000. 4 runs of their machine, 3 author paper, and 28-fold coverage. And if we wanted to speculate what would go down here with those next generation sequencing, people are speculating less than $1,000, maybe 1 run. I don't know how many authors they want to put on, that'll be subjective. But this is what's happened in the last 8 years. So it's every bit as dramatic as what I was showing with the total amount. Lewis?
What's the unit for calculating the coverage?
Oh, coverage is how many, basically if the genome is 3 billion base pairs long, essentially how many sort of reads are you overlapping any given position, which is a bit of a sense of how confident you are of what you've got there. Yeah, so I mean, you don't do it just once, you do it multiple times and then sort of stitch them together. Okay, so this is the paper that, this is the, editorial that came out in the October 2008 issue of Nature Biotech, Prepare for the Deluge. So this is where I got my title. And a couple more slides and then we'll have a pause. So of course we've got all this data, how do we store and retrieve it? Well, older people in the audience will remember things like this. Tapes that you put stuff on, and you may know what the capacity of tapes are. But let's look at hard drive storage growth. This is from the Wikipedia. Kilobyte, megabyte, gigabyte, terabyte, petabyte, and that's how it's going up over this period. So where it counted, it started going up exponentially. I mean, it would be very sad if we were trying to get the masses of data that we are collecting now in a period where basically disk space was not growing at a comparable rate. And here are some of the devices that older people in the audience will remember. Floppy disks, zip disks, well, CDs you probably still remember. This guy here, hard drives, are becoming more and more essential. The amount of data you get off one of these current machines is too large to pipe down, to download, say, remotely. So my colleagues who want to give me some data off their machine, they get on the tram and travel from where they are to my institute and hand me one of these hard drives. That is the most efficient and almost the only practical way to hand over, maybe half a terabyte of data. This is Moore's law here, and it's been helping us a lot. I don't actually have the capacity for the thing sitting on your desktop, but the supercomputer trend, number 1 and number 50, and the stuff on our desktop is growing at the same rate somewhere down here. So we've been relying on these things in both processing speed and storage capacity to feed what was going on back here. This sort of stuff. Okay, so this is where we've got to. Where are we today? Well, I'll just quickly show you where we are today with one of the technologies. Illumina sequencing technology starts off with millions, hundreds of millions of little sequence reads which are there that you want to read. They have stuff put on the end and they get popped onto a glass slide, which is not really, doesn't look very much like glass there, and I'm now cursing myself that I didn't, yeah, I've got it here. I bought one, or one or two to show you, but I have a horrible feeling. I left it in my hotel room. So I bought some of these Illumina slides, but unfortunately I forgot to get them in my rush this morning. Okay, so these guys here are put on this surface, and what happens is they're copied in the Illumina and the Applied Biosystems technology. They're not copied in the Helicos, and then they're read. So we've got these guys sticking up. They've got these special adapters to help that. And we want to read the sequence. And what happens is they're read from the top down. So we have all these guys here, and we're going to sequence one of these strands by adding on the complementary base one by one. So we add on the first one, we zap it with a laser, it emits some intensity of colour, and you pick that up over here in an image. So this image with these different colours are to reflect the colour that was basically emitted, or the wavelength of the light that was emitted when the laser hit the first base that was added as we go down each of these clusters. So every one of these is a cluster, and we have tens of millions of clusters on the slide, and this is a sort of a picture of a tiny portion of one, and we get a colour from the laser. And then you do it again, and you get another image, and another base is incorporated, and you keep going all the way down, you get a lot of images, and then you've got to read the images, so we'll take one cluster and follow it through, and we read off the bases. So that's very loosely how the Illumina technology works, and the Applied Biosystems and the Helicos are not hugely different, except the Helicos skips the clustering. It just does single molecule straight off. Whereas the next generation technologies that do single molecule are really quite different. Don't involve incorporation of bases in the same way. Okay, so we've got our sequence here now, and well, this slide is courtesy of Illumina, needless to say. And I just want to show you. Sorry.
Last slide, the bridge amplification, the middle slide, the band, the
Yeah.
bridge. Yeah, I saw the animation on the website, the Illumina website yesterday. I'm sort of confused with the bridge amplification. Yeah, that's.
Right, well, they have these adapters on each end, and the idea is, in some sense, they bend over and pair with the other adapters that are stuck on the surface. How can they become straight?
Yeah.
Well, you mean during the amplification?
Yeah.
Obviously. So I guess probably, what is the answer to that? How do we make them stand up in there so that we get like that? Don't know the answer to that, but I'm guessing we probably just have to add something which will break the connection down here. But anybody in the audience know the answer? Any? Oh, okay, sorry about that. I'm not the right person to ask, but anyway. Okay, so let's look at, this is something that Illumina presented to a talk I went to about a month ago, just to show how it's grown. This is 2009, starting, well, here's January 2009, and this is gigabases of quality data per run. And one run, as I'll show you in a minute, is one little, so it's not a chip, they call it a flow cell, a little bit like a glass microscope slide with 8 little things etched in it. That's one run. And it takes about 3 days. Okay, and the beginning of this year is 15 to 20 gigabytes per run. They're currently delivering 55 to 60, and they're on track to deliver 95 gigabases of DNA sequence per run. So that's almost a terabyte, or a terabase, sorry. And this is per run, let's say 3 days. Well, if we're generous, we do that once a week for 50 weeks. So we get 50 terabytes out of one machine per week. And already they've got, I think, 6 or more open at the Genome Institute. Okay. So, a little picture of one of these guys. I'm going to slow down in a second. This is the thing I was describing, which has the 8 lanes. And as you can see with those 2 fat fingers, it's not a very big object. And typically, the clusters that we had in our image are down those lanes. And here's a little bit of one. They're divided up artificially into 100 little pieces called tiles. And the analysis pipeline, I'm not going to go into this in any detail, but there's lots of mathematics and statistics and computation here. First, we've got to analyse the image. The raw image from one of these guys is about a terabyte of data. That's a terabyte, not a terabase. We're not going to get a terabase out of it yet. We're hoping to get maybe a terabase by the end of this year. But right now, it's a terabyte. And now, the images have got to be analysed, and then you've got to call the bases, and we might get 50 million, perhaps, and they have quality scores as well. And then these sequences, you have to do something with them, and if they're human, you're going to map them back to the human genome, find out what they look like, where they come from. If it's not a genome which has already been sequenced, then you have other problems, like assembling these small pieces, which I'm not going to discuss. But that's roughly where you get to. And that's where I'm going to pause and ask for a question or 3, because that's sort of the end of the technology stuff. That's where we are. I hope you'll agree. That looks like a deluge of data. If you're at the wrong end of it, it definitely feels like a deluge of data. But it's also, of course, a very exciting opportunity because we have lots and lots of new problems to deal with. But any questions about the technology that I might be able to answer? We just had one that I couldn't answer, but maybe there's one I could.
How is the compression technology cope with this?
So?
Compression? Data compression?
Yes.
So you compress it, it becomes smaller. Of course,
you're making a statement or a question? We can compress. DNA doesn't compress that dramatically. You know, if you want to take a gigabyte of DNA and then compress it, you won't achieve dramatic reductions. Because, you know, in a certain sense, from the statistical point of view, although there's lots and lots of exciting stuff inside there, from the, you know, let's say from zipping it up with a Lempel Z algorithm or something, it doesn't look very different from random sequence. I mean, it's just, it has structure at so many levels of so many different types that getting a compression algorithm to exploit that is really, you know, doesn't make a whole lot of sense. You know, so there are no huge projections to be made by compression. There are some, and of course, images and other things is another matter. But I'm talking about the actual genome sequence. And having said that, there are bits that compress very easily, but you know, there are sort of interspersed with stuff that doesn't. Yeah.
How large is this data set compared to the experimental data?
Good question. I don't know the answer, but I do know this. That astronomers go through their massive data sets to find the interesting stuff. Most of what we're generating here is regarded as interesting to somebody. So it's not like a small amount of interesting stuff in a huge amount of background. Like, you know, you'll point your telescope to some part of the sky and collect data for days or hours and then sift through it to find out the little bits. In a certain sense, every base pair of all of these sequences is potentially interesting for some problem. Of course, for most problems, we reduce it, try to solve the problem, and move on. But because of the fact that there's signal, in some sense, on many scales, of many, many kinds, there's no massive reduction. It's quite related to the question I just answered, right? If there was there was an easy way to find the stuff we wanted and throw out the rest, that would be almost tended down to a compression algorithm that just highlighted the stuff we want and kind of disregarded the stuff we don't. So, unfortunately, I don't know the actual numbers from astronomy or something else. But I assume that they are either as big or much bigger, but I think the density of information in the data is actually greater with the genome data. But I may be maligning the astronomers who love every little dot in the picture of the sky that they look at. But my experience is it's not quite like that. Yeah?
Maybe a related question, but in computer science, people have
once agreed that since we, for large problems, why don't we squash the data such that a smaller data set kind of represents the whole data. So that sort of data squashing, has that been done or have been looked at? So
Well,
basically the
I
idea
guess I don't quite know what you're saying, so I can't say yes or no.
So basically, instead of, since the data sizes are larger, instead of scaling our algorithms to deal with more larger data, there was one notion that you squash the data in such a way that a smaller data set kind of represents the whole thing and gives you kind of same similar answers.
I feel like saying, does anybody in the audience like to answer that question? Because I don't think I can.
Compressive.
It doesn't mean
It's
that's
not
squash.
exactly compressing, but it's basically squashing. You basically somehow you look at some things in the data which essentially represents the whole data.
Well, I don't know quite what that means, so I can't say yes or no, but what I will say, which is slightly related, I think it's also related to what I was saying to the answer to that question. When you collect this data, there are lots and lots of things that you might look at, but typically you collect it for looking at some particular purpose,
Okay.
and which already involves distilling it, summarizing it, and focusing on what you want. So you don't all the time look at everything that you could look at, right? And I'm going to give a little example later where we're looking for peaks of a particular kind, and so all of a sudden, 100 million little reads gets distilled down to something quite tractable and we deal with it. And then we just move on. Although in principle, we could have done many, many other things. But, time is short, life is short, so you collect your data, answer your question to the best of your ability, and get on to something else. So, but I guess the point is, we can't say a priori, we can't just collapse it a priori before you've decided what you're going to focus on. So in that sense, generic compression, maybe generic squashing, if I knew what squashing was, is probably not that applicable.
How about the gene of two humans? Would it be quite the same?
Two humans are reported to be about 0.1% different, about 1 in 1,000. But I don't know, with 3,000 million, that's still quite a few differences, right? So, and of course, finding those differences is challenging. And that would be in support of my argument that in principle, every bit could matter. Because if we're going to sequence you, and we're going to sequence me, and compare them, we don't want to compress them and lose any information. So it's not allowed to be lossy. We've got to keep it all there. And then we've also got to know enough about the structure of the data to be able to get gains. And that's where we can't do it, if we want to find exactly the places where you and I differ. That was your first question?
What I mean is, perhaps one part is the same.
Sorry?
For the two humans, this part and the other part is the same, so you just call one part and you say that.
Well, I mean, the way we actually compare two humans is by comparing them both to a standard, a reference human. Though that may increasingly be difficult. As we get hundreds of fully sequenced humans, the reference human may start looking a little bit unhappy because across many, many different humans, we may have things that aren't really illuminated by comparing to the reference. But in principle, for the single base changes, this is straightforward. Unfortunately, there are insertions and deletions, sometimes quite large ones, which are quite hard to describe and are not easily accommodated by simply comparing to the reference. Yeah?
You were talking about the sequence read, right? Read lengths from the sequencers.
Right.
In terms of the read lengths, would you prefer, say, a Roche machine that gives you a 400 base pair read to be better than, say, an Illumina 150 base pair read?
Well, that's a good question, and the answer is it depends. There are some tasks where more is better, and there are some tasks where, like for example, if I could give you 10 million sequences, would you rather have 10 million of length 25 base pairs or 5 million of length 50 base pairs? And the answer is for some problems, one is better, and other problems, the other is better. It's not like more is always better, because it does depend what you're going to do with it. For example, if you want to assemble, and for several tasks, the longer the better, or maybe you need things at different scales. Whereas the task that I'm going to describe in a few minutes, which is identifying the binding sites Actually, it's probably better to have more reads of a limited length rather than longer ones. At least the way I'm thinking about it. But I don't think there's any one answer to that question.
Just the reason I was asking is because you're talking about coverage, right?
Yes.
The shorter the read length, the more you should have coverage.
Oh, no. I mean, the coverage will be the same if you have twice as many sequences that are half as long. So the coverage is really just the average number that overlap a particular position, loosely assuming that they are uniform, which they're not, but let's pretend they are. So that won't be changing the number because the length and the number of reads will just trade off against each other. Okay, that was excellent to have some questions and break my monotonous voice. Is there another question or two before I move on? I'm sure we've got time. I'm rattling along and we're here all night. No problem. Okay, that's the end of questions, and if not, just magically moving the lights. Okay, so I want to touch on a question which is not my primary interest, but just to show you how we had to move along in algorithm development to keep up with this. So I'm going to give you a sort of kindergarten version of the problem of alignment. So if we have a sequence which is that, just a string of A's, C's, G's and D's, and we have another sequence which is that, the alignment problem is to find out whether this sequence is more or less the same as that, give or take a few little changes and maybe some insertions and deletions. So here is an alignment where that G matches with that G, that A matches. We've got a few perfect matches, and we have one mismatch, and we have 4 insertions. Right, so that's called an alignment because we've kind of put them under one another. Obviously, if they have different lengths, we're not going to be able to align them without some insertions or somewhere else. We could have had ones up here as well. Well, it turns out that is an optimal alignment if you score 2 for a match, -1 for a mismatch, and -2 for an insertion or a deletion. Trust me, it's true, I think. Okay, so that's what we mean by an alignment. You could get different alignments using different scorings. For example, that's an optimal alignment if I score matches 1, and I don't bother about mismatches or insertions and deletions. This is sometimes called the longest common subsequence problem, where we've matched them, see the G, A, T, C, G. That's a sort of common subsequence of length 1, 2, 3, 4, 5, but we can just fill in the rest with insertions or deletions. So there are 2 alignments. Finding alignments is important because we want to decide if we get new sequences whether they're really just copies of old sequences. So we go back to our database with our new sequences to see if there's any copy of it, and we have to do this sort of thing. These alignments were done by an approximately 40-year-old dynamic programming algorithm, and things have moved along a bit since then. Dynamic programs, I love the little procedure, and these algorithms are great, but they work fine for this sort of example, and they might even have worked fine when the length went up to hundreds and thousands. They did not work fine when the length went up to 100 million. So let's look at the history of sequence data growth, just to remind you. That guy there was 5,000 base pairs, 16,000, 50,000, 120,000, 1.8 million, and so on. So they're getting longer and longer. And as these genomes get longer, we have to do alignments that are longer and longer. And here's a sort of a corresponding development of algorithms. We've got to keep computer scientists busy in some way, get them off the street. Well, back here, Illumina and Smith-Waterman were based on the dynamic programming algorithm that I described, I mentioned a couple of slides back. After a while, the database, the GenBank database, got so big that even if you had just 100 base pairs, if you wanted to compare it with the GenBank, which you remember was going up at roughly a little less than 2-fold per year, these guys would not do the job. There was a time when hardware devices were made to implement these algorithms because just running them on a normal processor didn't work. So FastA and BLAST were developed to overcome this bottleneck around about 1990. So that's a new sort of quick and dirty algorithm, if you like, just to get the alignment problem solved because you're comparing one shortish sequence, maybe 100 or even 1,000, to, well, maybe something as big as a billion base pairs. Brilliant. And then, well, okay, a little bit later, hashing algorithms came in, and first off, they would hash the algorithm, hash the genome, and then compare the sequence to the hashed genome. And then, as the next generation stuff turned up, we started hashing the things you wanted to align to the genome. I'm not going to explain what hashing is and why this works, or even what all these names mean, but BLAST, for example, was used to perform the alignments that got the public human genome sequence together. So BLAST was certainly inadequate for that task. Up here, ELAND, MAC, they are the ones that are being used currently for the next generation sequences. And, well, somewhere up there we have Bowtie. BWA is meant to mean Burroughs-Wheeler algorithm, which is a fancy word that computer scientists rediscovered a couple of years ago, which turns out to be very helpful in this context. SOAP 1, SOAP 2. So the main point of this slide is just to show that as the technology grew, as the capacity to store the data and process it grew, we still had to have a corresponding development of algorithms. You couldn't stick with the simple dynamic programming alignment algorithms that were great back here, even though that may be what they're teaching you in textbooks. This is what they ought to be teaching you, because that's what we need right now. And of course, well, all of these things in between are still being used. There are still people using BLAST to, query databases. So, I'm now going to sort of wind up by talking about, I won't give you a timeline in this case, but some of the statistical problems that have had to evolve in the same sort of way. Unfortunately, they're not quite as neat as this, because statistics isn't as simple as, statistics isn't as simple as taking two sequences of letters and finding out how they align. So forgive me if I don't do the development quite as much. But of course, the mathematical, statistical, computational methods do have to evolve in the same way, but they tend to be driven by questions. And here's a collection of questions. This, needless to say, looks like it's taken from the company website that I can read you through. Over here, protein-DNA interactions. I'm actually going to talk about this one. Over here, temporal changes in expression, mutation discovery and profiling, populations, copy number variation, environmental exposures. So this is the environmental stuff that I was talking about. DNA barcodes, compound libraries, microRNA expression, alternative splicing, mRNA expression and discovery, metagenomic and heterogeneous samples, transcriptionally active sites. So these are all little problems within the world of biology that are being addressed using the next generation sequencing data. And my point is that all of these guys are going to need new algorithms to address the questions that arise in those contexts with this new sort of data. And it's not new intrinsically because it's DNA sequence data, but the nature of it and the quantity of it. Tens of millions or hundreds of millions of very short sequences adding up to maybe a terabyte in total for one run, one experiment. That's something that needs new algorithms. And, well, what am I going to do here? I'm going to talk about that, and I just wanted to do a little bit of advertising for the local team, because this is a paper that came out earlier this year, I think, 2009. Some of the best biology and associated algorithms on this particular problem of protein-DNA interactions is being done in your very own Genome Institute of Singapore, Agency for Science, Technology and Research, blah, blah, blah. And also the NUS graduate program, which I guess must be, who's in the NUS graduate program? Hugo's not, Ed's not, Melissa's not, must be him.
No,
Melissa is in the graduate, I'm sorry. I couldn't choose already a graduate. Okay, so this is a great paper, and there's another a better one in the pipeline coming from Nature. Okay, so what's the problem? One particular version of this DNA protein interaction problem is what's called ChIP-seq. And I have to tell you what ChIP-seq means. So we start off here with a cell and a nucleus, and we cross inside that nucleus we have our chromosomes, and these coloured blobs, which are proteins. And these proteins, the ones that bind to DNA, we're interested in finding out where they bind. So what's called cross-link. That means you basically glue the proteins that are near the DNA or binding to it, you stick it so they're stuck wherever they were. That's the cross-linking. And see, here we have now, and then we lyse the cells, which means you basically just burst them open and their insides all come out, and you get lots of bits of DNA with a protein stuck to it. So that stuff is called chromatin. Protein DNA stuck together. And it says isolate the DNA and fragment it. And that's what this is doing. And we have now little bits of DNA with protein stuck to it. Then you immunoprecipitate. It says have an antibody and you pull out the bits that do have protein there and you get rid of the rest. So we have little bits of sequence that were stuck to the protein by this cross-linking up here. So that's the chromatin immunoprecipitation. And now we need a seq bit. Well, we're going to sequence the little bits. We reverse the cross-links and take the DNA, select the size, and then sequence it. So, this is a way of finding out where the protein was binding to the DNA. Glue it together in the act of binding, as it were, break it up into pieces, keep the pieces where the protein was stuck to the DNA, then reverse it, getting rid of the protein, then you've just got the little pieces of DNA, then you sequence them and map it back to the genome. So this is taken from a Science article in June 2007, and I should just say that this paper here talks about doing this sort of thing, and here you have the old ABIs, we have the 454, the Illumina, and the ABI. What you don't have is the helicos, but I'm sure now that I've said that, I'll probably get one pretty quickly, only about 500,000. Okay, so I'm interested in proteins binding to DNA in points. That's a sort of schematic of a point. There are other binding events which are more extended in nature, and the story that I'm about to present won't be applicable to them, but I'm hoping some modification of it will be. So this is what we're going to be talking about for the next few minutes. Transcription factor binding, which we think of as a point event. So here's a little experiment that I've been involved in, just to produce some pictures to show you the idea. And I'm only using this to illustrate one approach to one novel problem prompted by this new source of data to address an old question. I mean, in the old days, if you wanted to sequence this stuff, you just send it off to your ABI sequencer. That was expensive, it was slow, and you didn't do a whole genome because you couldn't possibly afford it unless you were Craig Venter or some incredibly rich company. So, this experiment, we're involved in a particular cell line, which we won't worry about, called U2OS beta, and it expresses a thing called ER beta. So that's the protein that is going to bind to the DNA. We do the immunoprecipitation with an antibody specific to the ER beta. That's this bit here that pulls out the fragments where the ER beta is bound to the DNA. Now, in biology, you often have controls that show what happens if things are not as they should be, because sometimes you turn out that you do something and you think, oh, I got a great result, and then you do something which should have got the same result, and it does give the same result, and you have to sort of figure out what's going on. So in this story, we've got a control. We use a non-specific antibody that should not pull out the fragments that were bound to the ER beta, but it's going to pull out something. And then the DNA is sequenced in Illumina. And our interest is simply to find the genes that are close to a binding event. And I'm just going to very quickly sketch this. So here's a tiny bit of the data that was generated about a year ago, a little more than a year ago. At this time, the specific channel, this is one lane of one flow cell, gave 14 or nearly 15 million reads. Now, actually only about a third of those turned out to map nicely to the genome. So this was a rather stringent procedure. We'd like that to be much bigger, but still having 5 million little reads mapped to the genome is still not bad. And about the same for the non-specific. Now, these guys ought to be kind of randomly everywhere, not at all interesting, and these guys should be sort of piled up near the genes that the ER beta likes to bind to. And that's what we're interested in. So that's our raw data, if you like, two sets of a bit over 5 million reads. And a read in this context was about 27 base pairs long on average. So we have, let's say, 5 million, 27 base pair fragments coming from somewhere in the 3 billion base pair genome. Now, the story that I want to describe superficially is, here's the protein binding to the double-stranded DNA, and each of those ends, this is, because they get labelled, and we know which end they come from, and the guys that are on, as it were, the left end there, these guys, this is called the forward strand or the positive strand. They're going to map to one end of the site, because this is actually the site in the middle, and these guys are going to map to that end. So it turns out that the ones that map to the forward strand are slightly offset relative to the ones that map to the reverse strand, and that diagram is meant to suggest why. If we actually look at some data, we do see this sort of thing. So now I have 1,000 base pairs from somewhere in the genome, and I put a little dot where the left end of one of the reads maps. If it's the black dot, it's the left end of the forward strand. If it's red, it is the left end, which is going the other way, so it's the right end of the reverse strand. We put the dot there, and you can see the black dots. Firstly, you can see they kind of build up in a somewhat triangular shape, and the red dots are offset to the left relative to the black dots. So that's one depiction of the raw data that would suggest that wherever this spot is in the genome, there was a pretty strong binding event at that position. And the task is to analyse the data to find all those binding events in the whole 3 billion base pair genome. And that is an entirely novel task in the sense that before these sequencing machines became available, no one ever dreamt of trying to solve this problem. These guys here, that's probably not a binding event, that's sort of background, but we don't know for sure. So there are a few aspects of the data. Now the first idea might be, apart from binding sites, the reads should be roughly uniform. I mean, if you've got all this stuff, there are binding sites where you should have a cluster of reads, and then there's the other stuff, and you might think it's wrong. Well, it turns out to be wrong in an expected way. Not every read can map to the genome, because the genome has a lot of repeats. Almost a half of the human genome is what's called repetitive. And if you've got a sequence and it exists, let's say, 500 times in the genome, then it's not very easy to map it somewhere. And most of the mapping algorithms at this point don't do anything with sequences that map to multiple places. There are ways to try to deal with that, but the standard Illumina story throws them away if it maps to multiple places. That was why my 15 million reduced to 5 million, that there are these issues of mapping. They were unexpected. There was a strong bias of the GC content of the reads. There were also too many read-rich regions, and so there are a few technical issues. It wasn't quite as simple as we'd liked. And here's the little picture, which I don't want to go through in detail, but just to show you, even though this is really great data, there are artifactual issues that need to be dealt with. GC content is along the bottom here. That's simply the percentage of Gs and Cs in the DNA sequence. And I didn't mention back at the double helix level, but the Gs and the Cs are a stronger bond, the 3 hydrogen bonds, than the A's and the T's, and this has implications for many of the processes involved in dealing with DNA sequence data. And the GC content tends to vary in a slow and non-random way. So there'll be some largest regions of the genome with very high GC content, then it'll move to regions with lower GC content, and it sort of fluctuates in a very complicated way. But typically, if you have 1,000 base pairs, it'll have a GC content which is characteristic of that region, which might be high, might be middle, might be low. And this picture is supposed to display the heterogeneity in the number of reads that will actually just go there, that will just map there. So it's probably something to do with the actual process of generating the data, but nobody seems to really know exactly what's going on here. Okay, but now let's suppose we take, these are 5 kb, so let's take all little bins of 5 kb and see how many reads map to it, and that's almost a perfect Poisson distribution. There's a bit of a problem at 0, and there's a few guys at the end here. This is background non-uniformity. So this is the background where nothing interesting should happen. If we do the same thing with the foreground, with the specific antibody up here, with the going. The specific antibody there and the non-specific, they differ very little. There's a bigger hide bin there. These are a number of reads in 1 kb bins, it says there. So 0, 1, 2, 3, 4, and here 10 or more. And there's more guys there, and then there's still a pile of 10 or more there, many more than you would expect by the Poisson. And this has high noise and high signal, this just has high noise. So the stuff we're interested in is in there. And part of the game is to find the places where there's something interesting and then zero in on it. And I guess this also illustrates my point that although we have maybe 10 million little reads, with lots and lots of information, for this purpose of addressing this question, we've done a pretty drastic simplification already. We're bidding 1 kb regions and then going in and looking at 1 kb regions very carefully. So, we do it in two passes. We identify the enriched regions by just moving along with our 1 kb, and then we fit a shape that looks roughly peak-like, and we check how well it fits. And here's a little bit of math for my shape. We're going to model the positive strand, or the 5' end, to be some shape, S, and T is the position along the genome, and A is an amplitude, B is the centre, so this is the shape, and position minus centre divided by scale factor there, plus background. On the positive strand and the negative strand is the same story, except that we've got a little offset. So this is a very simple model for the Poisson rate at which the reads are landing around the peak. Now, if you ask why is it Poisson, well, we have to look at where the Poisson fits, but it does remarkably well, actually. So we're basically going to try fitting that model. But so my shape model that I'm using is a simple triangle, and here's an indication that it more or less fits. This is the data I showed you before, and if you smooth it a bit with a kernel smoother, you can see we do indeed have something approximating two triangles with a bit of an offset. So we're going to inflict that on our data for every bin which seems to have too many reads to see whether the reads fit this shape. And if they fit the shape, we'll say, yeah, that looks like a possible binding site. And if they badly misfit it, we'll think it's some sort of artifact. And we do the fit using a very traditional technique called maximum likelihood in statistics, where we get the Poisson likelihood and we maximise it over those parameters A, B, C, D, and E. So, and we do that and we get our story. And in the data I showed you, there are about 12,000 bins of 1 kb that have reasonably convincing triangular or some other sort of shape. And we have some further problems of deciding how good the shape is. And well, here I've just given an example that sometimes we'll have two of these triangular looking objects in the same 1 kb bin. So we actually have to modify this story to fit multiple peaks in the same region. But most of the time, 1 kb will only have one nicely defined bin. So that's my illustration of the sort of new, but not terribly new, statistical ideas that you can apply to this completely novel set of data to address this question of identifying the triangular-shaped binding sites. If I go back to this slide here, we have all these different problems, and every one of those problems with this new source of data are providing novel analysis challenges. So this is what the deluge has brought upon us. Lots and lots of data, lots and lots of challenges to address the data, and there's no doubt this list of questions that people are using to answer with these data is going to grow as the amount of data grows. With that, I'm just going to close with a quote that I like very much. In a way, I wouldn't be in any business at all if things didn't change. Because if you're just developing the same old techniques for the same old data, it gets stale very quickly, and you have to work very hard. Because doing better on old problems is a lot harder than doing something simple on new problems. So I feel, go for the new stuff, folks. So I'm indebted to Heraclitus for saying this very nicely. It's also true for technologies, for applications, for the math, stats, and the computer science. And here are the people that have helped me with this. Oleg is the student in Berkeley who's working on the ChIP-seq on the binding sites. Had a lot of input with Illumina people, not just pretty slides, but we've had scientific interaction. And these are my colleagues at Lehigh, all of whom are working on some aspect of next generation sequence analysis. And that I'll thank you for your attention.
But before Professor Speak answer the question, or he's not hungry, I would like to present an appreciation to him on behalf of the institute.
I'm sorry I went over time. I guess we started well, maybe a little after. Yeah, but anyway, I'm happy to answer questions. Anybody has one? If they don't, I'll be disappointed. I'll have to commit suicide. Oh, definitely. Yeah, sure. I mean, well, he correctly points out there are already about 9 algorithms out in the literature to solve the problem we're working on. None of them are doing it quite the way we're doing it, and of course we hope to do better. Otherwise there's no point in it. And I should say that there's also a very good algorithm from the Genome Institute of Singapore. So yeah, this is definitely a competitive area, and I'm interested in whether the parametric model that I'm fitting can be used to give a more discriminated selection of which overrepresented regions are really binding sites and not just some artifact. Because sometimes artifacts look a little bit like binding sites. But when you want to find a nice triangular intensity with an offset that is quite reasonable in relation to the length of the fragment, and some of these things, we hope our parameters can be used to filter out the bad stuff. That's the hope. But if it doesn't work, too bad. We'll go on to something else. But yeah, there's plenty of algorithms already. And we use those while we're developing this. Yeah?
Yeah, so you seem to be a master. So one of the problems with these kinds of data are that we don't really know the truth very well. We can kind of have some idea of the truth, but we can't find out the whole truth.
Correct.
And you seem to be a master at dealing with these kinds of situations. So what advice can you give us dealing with these kinds of data where the ground truth is incompletely flipped?
Well, I decline to agree to any of your statements. But you do have to do your ingenious best to make up pseudo-truths or near-truths or half-truths or something. Of course you can simulate, which is okay, but what about down on the ground? Right, so, yeah, so we do spend a lot of time trying to get some way to test the efficacy of your algorithms. I mean, if you ask me what we've got, no one has genome-wide binding truth. You can, of course, select things, and people generally select the best-looking things and do some slow chip analysis. But it's generally unrealistic to, if you've got 12,000 predicted peaks, to check a reasonable fraction of those. So we have to do other indirect methods, right? I mean, one of them might be the motifs. I mean, there's sort of a range of indirect methods. You missed my talk this morning where I gave a kind of milk donut. It was a very good move. But we've invented some, maybe not very convincing, but slightly convincing ways of checking copy number segmentation. Because again, when you've got copy number variation, there could be hundreds or thousands of places where you have aberrations. How do you check whether your algorithm is delivering the right answer? So, I think the answer is you just try to invent something. Yes?
GC rich sites tends to be like real chromatin region, right? They tend to be like open.
Right, yeah. Well, open chromatin is another issue. That's part of the problem.
So I just wonder, like, for example, in the binding sites that when you see your leads, do you observe that they are tend to be GC rich regions?
Well, of course, but that's partly that promoters tend to be more GC rich, and the binding sites that we recognise tend to be in promoters.
Then how
But
do you actually separate that from the artifact of
Well, the short answer is we don't know how well we do that. Because the peaks you find tend to be more GC, locally high GC rich. And we select our bins as candidates for peak examination by looking at whether they are more than what we would expect accounting for the GC content. Now that may not actually be a good idea, and that's one of the things that we have to check very carefully. Yeah, so I think the short answer is we don't know if we're dealing with this adequately at the moment, but it could be an issue. Yeah.
You were talking about looking at those transcription factor binding sites, right? So, have you accounted for any polymorphisms in such regions, like say, because when you're talking about diseases, when you have polymorphisms,
Oh yeah.
that might be actually be the difference between say a normal person and a diseased individual.
Of course. Yeah. I would say probably at this point, this is a little crude, but of course if, for example, you have a nucleotide variation in the middle of a binding site, then it may not bind, and me it might. But we're a long way away from finding information about binding in you or me. We could take a little bit of tissue from you and a little bit of tissue from me and do a ChIP-seq, and maybe, if we're lucky, we'll find a few sites where I bind and you don't, or the other way around, and we examine underneath the site and we see that the sequence there is different between you and me. I mean, that'll probably happen at some point, but I have a feeling at this stage this is a little crude to be doing too much of that, but that will happen in due course.
So because we have maybe bioinformatic predictions which do that from a DNA sequence point of view, right?
Yes.
Like for example, if I say that this polymorphism that occurs at this position, it might lead to a change in the transcription factor.
Absolutely.
Right?
Oh yeah.
So if I need to say come up with a hypothesis and if I want to prove it, will your algorithm be able to account for it?
Well, let me say one reason why I think it's a bit crude at the moment. Firstly, these ChIP experiments seem to give much more binding than appears to be meaningful. Like, it's not reflected in gene expression, right? So we don't really know that too well, I think. And what was the second point I wanted to make? Even things that have very well-defined binding sequences, when we look under the peaks that we got, we don't always see those guys, right? So that you can have, think this transcription factor has a fantastic binding sequence, very canonical. Everybody believes that's where it binds. And suddenly I've produced you 5,000 peaks where there's not one there. So, it's hard to know what's going on there, right? We've got binding, we don't have any consequence of it that we can see. We have apparent peaks that don't relate to what we think are the important binding motifs. We'll sort this out eventually. But, there may be some artifacts, there may be some new biology.
Do you have some algorithms which allow you to improve situation with respect to discrimination, physical binding sites and functional binding sites?
Well, it's related to the question I just mentioned.
It's essential for this situation because sometimes again we see a very strong peak, but it is completely false in context of the functional activity.
This is a question on our agenda.
But mathematics and statistics, is it possible to move in this way without additional experiments?
Well, here's an obvious thing you can do. Well, you need some more data. You need some expression data. Right? And so, for example, that ER stuff that I talked about.
Without additional expression.
Without expression. See, with expression, you can say, let's look at the binding in the genes that we actually saw expression, let's say expression differences, right? If I do, for example, stimulated versus unstimulated, and I have binding in both cases, and I have expression in both cases, I can then look at the peaks, the shape of the peak, the sequence underneath the peaks that correspond to sites associated with genes that did something and associated with genes that didn't do something. And we did see differences, of course. The ones that are associated with genes that did something tend to be stronger. And of course, tend to be more likely to have the motifs. But you want to do it even without expression data. I think I'll leave that to you. That sounds tough.
Well, I'm sure Professor Speed will go on
two questions. But
He's hungry.
But I think he is hungry.
I'm hungry for discussion.
Okay. No, he won't realize.
Maybe you find it on scale.
No, he won't realize it, but I think he is hungry. Anyway, I haven't heard so many lectures that poses so many challenges, right? And I think yet Professor Smith has given us such a lecture. So please join me in thanking him for stimulating and very interesting